Hello everyone. Today I will give you an introduction on alcohol oxidases. I will start with a general introduction and then talk in detail about representative members that belong to the copper radical alcohol oxidases and the flavoprotein alcohol oxidases. I will talk about their occurrence in nature, enzyme activity, which includes the mechanism, and their use in biocatalysis in industry. And then I will end with a conclusion and a future outlook. So these are the reviews I found very, very helpful, and most of my research is based off them. So if you're interested, have a look. So let's get started. Oxidations are a cornerstone in organic chemistry as they give an access to a range of functional groups. And chemical oxidation methods typically require harsh conditions and pollution reagents, and they often suffer from poor chemoselectivity and enantioselectivity. Nature, on the other hand, has come up with a versatile set of enzymes performing a wide variety of selective oxidation reactions. So to get into biocatalytic oxidation reactions, mechanistically, biocatalytic oxidation reactions fall into two categories: dehydrogenation and oxyfunctionalization reactions. In the dehydrogenative case, an acceptor molecule, it's mostly nicotine amide or flavin cofactor, abstracts a hydride from a heteroatom substituted carbon atom. Here you see lacases and peroxidases that catalyze hydrogen atom abstraction reactions, mostly as you see on phenols, and this results in phenoxy radicals as primary products, and then they can react further. Alcohol dehydrogenases and imine reductases are often used in reductive processes, and oxidative applications are far less common. And then finally, oxidases, they catalyze electron transfer onto molecular oxygen, yielding either hydrogen peroxide or water. Oxidases include flavoprotein oxidases, such as alcohol, but also amine and nicotine amide oxidases. And then there are the metalloflavin oxidases and heme protein oxidases. So for more detailed information on the different enzyme classes, please have a look on our YouTube channel, where my colleagues also present other enzyme classes. But now the focus will be on alcohol oxidases that convert primary and secondary alcohols to aldehydes and ketones. The reaction is irreversible as hydrogen peroxide is generated. In order to avoid enzyme deactivation, for example, a catalase here is usually employed, particularly on preparative scale. In contrast to, for example, dehydrogenases, oxidases do not depend on a coenzyme that need to be regenerated, but they rely on molecular oxygen as electron acceptor. Alcohol oxidases contain either a copper or a flavin adenine dinucleotide cofactor. Compared to dehydrogenases, oxidases are much less abundant in nature and are often of eukaryotic origin. This can actually present challenges in terms of the development of suitable, efficient overexpression systems. So carbohydrate oxidases I will talk about are found in all kingdoms of life, but are mostly found in fungi. Flavoprotein alcohol oxidases are found in algae, bacteria, yeast, and plants. They are probably less abundant due to the fact that they produce hydrogen peroxide, and it is a reactive and toxic molecule, and also more damaging reactive oxygen species such as superoxide can also be formed. But the generation of hydrogen peroxide also offers a weapon to compete against other organisms through oxidative stress. In fungi, extracellular alcohol oxidases produce hydrogen peroxide, and this is also needed for lignin degradation by peroxidases. From a biocatalytic viewpoint, alcohol oxidases are a promising group of enzymes because they are biochemically well characterized, and a broad range of enzymes have been described, which were also employed in cascade reactions, as we will see. Their ability to work without any coenzyme regeneration make them cost-effective and less complicated, and therefore also more suitable for industrial applications. They find bioanalytical, agrochemical, and pharmaceutical applications. So let's get started with carbohydrate oxidases. Among enzymes acting on sugars, carbohydrate oxidases have been extensively studied and explored for their possible application in biotechnology. The physiological role of many carbohydrate oxidases is still debated, as the oxidation reaction often overlaps with the chemistry performed by carbohydrate dehydrogenases. Oxidation is typically directed towards specific positions of the sugar ring, as you can see here. Most frequently observed radioselectivities for carbohydrate oxidases are carbon 1, carbon 2, and carbon 6 oxidations. Such radioselectivity is difficult to achieve by chemical means. So several different classes can be distinguished. A first distinction can be made based on the cofactor. So we have the carbohydrate oxidases, and then they contain either a copper as redox cofactor or a flavin cofactor. Copper-dependent oxidases, they make use of a single copper ion, which is bound in the protein core, held in place by the surrounding residues. The best known member of this class is the fungal galactose oxidase, I will talk about in a minute. And all copper-containing carbohydrate oxidases belong to the same structural family. Flavoprotein carbohydrate oxidase can be classified according to the structural fold into two distinct families, the glucose-methanol-choline oxidases and the phenyl alcohol oxidase family. And the further distinction is made on the number of covalent bonds. So you see no covalent bonds, or a single covalent bond, or two covalent bonds. So yeah, let's start with copper alcohol oxidases. So galactose oxidase is the best studied enzyme of the copper radical oxidases. It is known since the 50s and secreted by different fungi. The biological function is generally unknown, but the natural enzymes are selective enzymes that act specifically on position carbon 6 of galactose, as you can see here. Other substrates include dihydroxyacetone, benzyl alcohol derivatives, and also galactose-containing polysaccharides. The structure of galactose oxidase corresponds to a monomer composed of three distinct domains. The N-terminal domain contains a carbohydrate binding module used to recognize the substrate, and the central domain that you see here contains the active site with the copper ion. The copper ion interacts with four residues, two tyrosines and two histidine. A unique and typical feature of this galactose oxidase is the presence of a crosslink that you can see here between a cysteine and this tyrosine 272. Such bond is formed post-translationally and is necessary for the reaction cycle of galactose oxidases, as we can see if we look at the catalytic cycle of a copper-containing oxidase. So since only a single copper ion is found in the active site, it seems surprising that a two-electron transfer can occur. And this is explained by the presence of a tyrosyl radical unit in the protein active site. It acts as a second redox center during the catalytic cycle. So here in A, the first half reaction involves proton transfer from the oxygen 6 from galactose to the acyl tyrosine that you see here. And this is an anion. And hydrogen atom transfer also from carbon 6 of galactose to the tyrosine cysteine radical cofactor, and electron transfer from the carbohydrate to generate then the aldehyde and copper 1+. And then, of course, to complete the catalytic cycle, a second half reaction as proposed to involve an inner electron transfer from copper 1 to oxygen to yield a superoxide to produce then a metal-bound hydroperoxide, and also a proton transfer from the acyl tyrosine to hydroperoxide to produce then hydrogen peroxide. And yeah, and then we have again the copper 2+ radical state of the enzyme. So galactose oxidase has a restructured substrate scope only on primary alcohols. And so there was a need to expand it for broader application in biotechnology. And Arnold and colleagues were the first to investigate galactose oxidase engineering by direct evolution. And they used random mutagenesis of the entire galactose oxidase gene. And mutant libraries were screened with the most active identified in each round. And this led to variant M3. And here you see the galactose oxidase variant M35, which used the M3 variant as template and was previously engineered by Turner and colleagues. And this one has been shown to oxidize a number of primary but also secondary alcohols. In all cases, the enzyme displayed a R selectivity, leading to the production of enantiomerically enriched S alcohols. Here you see indicated the EE of the recovered alcohols. And they have also developed a rapid and sensitive screening method for determining the enantiomeric purity of a chiral secondary alcohols using this enzyme variant. Galactose oxidases have been used in a number of enzymatic cascades for the synthesis of high-value value chemicals. And mostly they're used in the first step. And I've depicted a publication here in Science, which is actually the synthesis of a nucleoside analog islatravir, which is the HIV reverse transcriptase translocation inhibitor. And here in the first step, so they have developed variants of galactose oxidase that chemoselectivity formed this monoaldehyde. The galactose oxidase variants favored formation of an undesired S enantiomer, but they wanted the R enantiomer. And the S selectivity is consistent with the stereochemistry of a natural substrate D-galactose. But they had a variant in hand, which is variant F2, that showed relaxed stereoselectivity. And this suggested that its enantiomer preference could be reversed. And that's why they used this variant F2 as a starting point for evolution. And then they engineered an oxidase, this one here, you see here, and this displayed an 11-fold improved activity and a reversed selectivity. So not only with this mutation, they could also improve protein expression, stability, and activity, and achieved 97% enantiomeric excess. And an unpredicted catalytic promiscuous activity of the copper-dependent enzyme galactose oxidase was also reported. This one I found really interesting. So the enzyme catalyzes the one-pot conversion of alcohols into related nitriles. And this really under mild reaction conditions in, as you see here, ammonium formate buffer. So ammonia is the source of nitrogen and dioxygen is the only oxidant. So this is a green method. And the substrate group of the reaction included benzyl and cinnamyl alcohols. And you see the products formed here on the right side. Given is the analytic yield and the turnover. And I also did preparative scale synthesis with this compound 4C, where they achieved 75% yield. So there's always research for novel copper radical oxidases, of course, and genome mining has become a routine for efficient enzyme hunting. So through this method, also new alcohol oxidases were discovered. And through the analysis of this fungi genome, you see here, 2 new copper radical alcohol oxidases were identified and then investigated. And you see that they show a really similar catalytic site compared to the wild-type galactose oxidase and this variant M35 I talked about. But it showed a broad and unique substrate range, catalyzing the oxidation of over 20 aliphatic and aromatic alcohols with really high catalytic efficiency. So now I will talk about the flavin-containing oxidases. And first I will talk about flavins in general. So flavin-containing oxidases contain flavin mononucleotide or flavin adenine dinucleotide as redox factor. In only very few cases, flavoprotein oxidases contain one or more additional cofactor. Both are synthesized starting from riboflavin, vitamin B2. So phosphorylation of riboflavin by action of a riboflavin kinase results in flavin mononucleotide, here marked in green, and adenylation of this flavin mononucleotide yields then in flavin adenine dinucleotide, which is the most common flavin cofactor in flavoenzymes. Flavins are extremely versatile molecules that are capable of undergoing oxidation, reduction reactions by exchanging either 1 electron or 2 electrons. Flavins can thereby exist in an oxidized, 1 electron reduced, or in a 2 electron reduced state. In the majority of flavoenzymes, the cofactor is tightly but non-covalently bound. So talking about the catalytic cycle of flavoenzymes, They typically comprise of two half reactions. In the reductive half reaction, the oxidized flavin is reduced by the first substrate, and in the second half reaction, the reduced flavin is reoxidized. And the ability to swiftly react with molecular oxygen to reduce it to hydrogen peroxide, this sets flavoprotein oxidases apart from other flavoproteins. The oxidized flavin is regenerated by stepwise single electron transfer via the oxidative half reaction, which requires triplet oxygen, as it is a spin-forbidden reaction. Dioxygen acts as single electron acceptor and forms a superoxide stabilized by a positively charged histidine residue here. And then another single electron transfer yields a covalent hydroperoxyflavin intermediate, which eliminates hydrogen peroxide, and then we have the reformed oxidized flavin. So classification. I will talk about two classes of flavoprotein oxidase families, but you see there are more. But the focus here will be on the GMC family and on the valine alkyl oxidase family. And let's start with the GMC family. So here, glucose oxidase catalyzes the oxidation of D-glucose to D-gluconolactone and is produced in fungi and insects. The narrow substrate acceptance is really in line when you look at the structure of glucose oxidase, which revealed a rather occulted active site. Its main natural roles are related to the production of hydrogen peroxide. And its physiological role as a weapon against other also microorganisms has been proposed. And also because it's really represented in fungal species, an association with lignin degradation is suspected. It has countless applications in the food industry. For example, in the baking industry, glucose oxidase could be employed to improve the bread texture as alternative to potassium bromide, which could be cancerogenic. And also, already 30 years ago, glucose oxidase was recognized as an ideal enzyme for biosensors applications thanks to its stability. So next enzyme is methanol oxidase, which is referred to as the alcohol oxidase. It catalyzes the first step of methanol catabolism in methylotrophic yeast species, namely its oxidation to formaldehyde. And in its major active form, alcohol oxidase is a molecule of really high molecular mass that consists of 8 identical subunits, and each of them carry a non-covalently bound flavin adenine nucleotide. The crystal structure was elucidated only recently, and in vivo, the protein is compartmentalized into special cell organelles termed peroxisomes. And as alcohol oxidase is a really abundant protein, and its synthesis is strictly regulated and occurs at the transcriptional level. Methanol is its natural substrate, but in vivo experiments have revealed that alcohol oxidases can also oxidize short aliphatic alcohols such as ethanol and propanol. And it has also application in biosensors. Only recently an alcohol oxidase was characterized, which is actually a discovery of a glycol oxidase. And why is this so, let's say cool? Because glycol constitutes an abundant byproduct of biodiesel manufacture. And there's a growing interest in increasing the sustainability for this process through valorization of glycerol. And then there are engineering studies, and that resulted in the new variant of this glycerol oxidase with a new substrate scope that could be really broadened. And the attention was directed to diodes, which represent a really industrial important class of chemical compounds, first because they're really cheap, and they can be transformed into either lactones or hydroxy acids, which in turn are building blocks for biodegradable polymers. And except for the focus on diols as substrates, they also tested other alcohols and also secondary alcohols and amino alcohols, as you can see here. But the really most striking discovery is the ability of this variant to selectively perform double oxidation on one of 2 hydroxy groups for a subset of diols. Yet that comes to the mechanism of overoxidation, because the oxidation of a primary alcohol catalyzed by a flavoprotein oxidase does not necessarily stop at the aldehyde stage, but may also further proceed to the corresponding carboxylic acid. The second step is mechanistically less investigated, but the actual substrate has to be the aldehyde hydrate rather than its carbonyl form, because hydride abstraction yields a double resonance stabilized oxycarbenium cation, as you can see here, which upon expulsion from H plus gives then the carboxylic acid. In contrast, hydride extraction from this carbonyl form would lead to a really highly unstable, this is now hypothetical, acylium cation, which would be quenched by water molecule. This overoxidation of alcohol to carboxylic acids has been observed for choline oxidases, I will talk about in a minute, but also about in other oxidases such as aryl alcohol oxidase and hydroxymethylfurfuryl oxidase, I will also talk about. So choline oxidase catalyzes the oxidation of choline to glycine betaine, a compatible solute that accumulates in pathogenic bacteria and plants so they can withstand osmotic and temperature stresses. The enzyme is a homodimer with each subunit containing now covalently bound cofactor, and this is linked to a histidine 99, and here you can see the covalent bond. And based, however, on the alignment of amino acid sequences, choline oxidase has been grouped to the GNC family. And choline oxidase is unique in that it carries out a 4-electron oxidation of the alcohol substrate to then the carboxylic acid. And substrates are choline, but also analogues. Next enzyme is cholesterol oxidase, which is able to catalyze the oxidation of cholesterol to cholesterol. Cholesterol oxidases have been found in several microorganisms, where it has a role in the initial step of cholesterol metabolism and also in pathogens, where it helps to alter the physical structure of the cell membrane. Cholesterol oxidases with the cofactor now non-covalently bound are part of the GNC family, but those who have the cofactor covalently linked belong to the vanillin alcohol family. The conserved histidine is not the catalytic base that deprotonates the substrate. It's glutamic acid. You see here, this 361, and this acts as catalytic base. It can be used also in the biosensor field, but also for the production of steroids, and it is also used in biomedical application to treat bacterial infection because of its ability to damage the cell membrane. Now we will talk about aryl alcohol oxidases that are detected in several fungi and involved in lignin degradation. So white rod fungi secrete a bunch of oxidative enzymes participating in the degradation of lignocellulosic biomass, is a central step in carbon recycling. And peroxidases responsible for this lignin degradation, they require sources of hydrogen peroxide. Thus, fungi produce oxidases, for example, such as aryl alcohol oxidases, to produce that hydrogen peroxide. This enzyme family is accepting a variety of aromatic alcohols as substrates. Unfortunately, the active site is buried under hydrophobic amino acids, as you can see here with the phenylalanine. And as a result, substrates bulkier than primary aromatic alcohols cannot easily be accommodated by the wild-type enzyme. But a lot of engineering and mutations in the protein sequence have influenced that, and it have variants with broader substrate acceptance, and also this positively influenced gene expression of the enzyme. Aryl alcohol oxidases are used to support reaction catalyzed by peroxidases and peroxygenases. They're used for building blocks for chemical synthesis and flavors, also photosynthesis. And they are used in multi-enzyme cascades to produce bio-based precursors for polymers. And I've got some examples. So here on the left side, you see the cascade reaction for synthesis of dextrofan, an antitussive agent, and a collection of secreted functional unspecific peroxygenases, aryl alcohol oxidase fusion enzymes were designed. So these two enzymes were chosen on terms of their cooperative activity, which is really cool. And in case Here, the unspecific peroxidase is playing the leading role as the oxyfunctionalization partner. So this dextrofan is formed through oxygen dealkylation, and the aryl alcohol oxidase is playing only a supporting role, just generating hydrogen peroxide by oxidizing different, as you see here, aromatic alcohols. Next, some valuable chemicals obtained from lignocellulose are primarily furfural and is hydroxymethylfurfural. Much attention has been paid to its oxidation and reduction because it provides convenient synthetic pathways for the production of chemical building blocks for the polymer industry. One of these important building blocks is furan decarboxylic acid, which originates from the oxidation of HMF. And it can co-polymerize with diols, producing polyethylene furan decarboxylate, and among also other polyesters. And these could replace polyesters based on fossil resources, which are not degradable. Here you see in combination with non-specific peroxidase, but here also this is a galactose oxidase. Aryl alcohol oxidase was used to produce furan decarboxylic acid, but no oxidation of formyl furan decarboxylic acid, no, it's just formyl furan carboxylic acid, to this furan decarboxylate acid was detected when only the wild-type aryl alcohol oxidase was used. So it stopped at this stage, and a non-specific peroxidase was necessary, till the Banta variant reached 3% of furan decarboxylate acid after 2 days of reaction, starting from HMF. So, but there's also an HMF oxidase that was found in bacteria and shows activity for primary alcohols, primary thiols, and hydrate aldehydes. Secondary alcohols are not converted, indicating that the enzyme is very specific concerning the substrate type, but HMFO is promiscuous concerning the side chain next to the alcohol group. And most, but not all, of the identified substrates are aromatic, including furanic, phenylic, and cinnamolic alcohols. Diekmann and Freie described HMF oxidase, which was expressed in E. coli and oxidized HMF to formyl furan carboxylic acid. So again, to this stage, this was the main product, and only minor amounts of this furan decarboxylic acid were formed. But recently, 2 other HMFOs from the other bacteria strains, both heterologically expressed in E. coli, were shown to catalyze the oxidation from HMF to up to 99% of this furan decarboxylic acid after 96 hours. So now we come to the last family, which is the vanillyl alcohol oxidase family. Members of the vanillyl alcohol oxidase family have been found to be able to produce a variety of high-value aromatic compounds. As the name suggests, vanillyl alcohol oxidase can oxidize vanillyl alcohol to vanillin, but is also active on plant-derived allylic phenols, such as eugenol. Well characterized is this vanillyl alcohol oxidase from the fungi Penicillium simplicium, and the proposed physiological substrate is 4-methoxymethylphenol, which is demethylated to hydroxybenzaldehyde. Vanillyl alcohol oxidases share a common fold, which is different from the one I described from the GMC family. The general structure is once more divided in a cofactor binding domain and a substrate binding domain. And the cofactor is deeply buried in the protein structure, but in this case, it is always covalently bound to a cysteine residue. And often there's a second covalent bond between the cysteine and the flavin cofactor. Here you see the tyrosine arginine triad, which crucially is involved in substrate binding. And this is the cause of the selectivity of this enzyme. Then there's eugenol oxidase. So the genome of Rhodococcus strains contain remarkably large number of genes coding for oxidative enzymes. Among them, one gene codes for this eugenol oxidase that displays significant sequence identity to the vanillyl alcohol oxidase I was described before. And it has a similar but not fully overlapping pattern of substrate preferences. This oxidase is also active towards vanillyl alcohol, but displays higher efficiency for eugenol, converting it into conferryl alcohol. And what it is really nice, unlike vanillyl alcohol oxidases, which expresses really poorly in E. coli, eugenol oxidase can be produced in large quantities, and this makes it to an attractive target for large-scale oxidative transformations. It can also catalyze the dehydrogenation of various phenol ketones and the selective oxidation of racemic secondary alcohols. So the vanillyl alcohol oxidase family contains various more enzymes that perform mostly alcohol oxidations in the production of aldehydes, ketones, or lactones, but they are also capable to perform amine oxidations, hydroxidations, and ether bond cleavage reactions. And this includes also an oxidase capable of CC bond formation. And one known example is reticulin oxidase, also known as the Bourbon bridge enzyme, which is involved in the biosynthetic pathway of plant isoquinoline alkaloids. So with this, I'm at my conclusion. So we've seen impressive progress in the discovery of new copper radical alcohol oxidases and also flavin-dependent alcohol oxidases. The application range from their use as biocatalysts for the synthesis to the integration in biosensors. And we have also seen useful cascade biotransformations. Then there's a really improved substrate specificity, catalytic selectivity, and stability through bioinformatic-based genome mining, especially for copper radical alcohol oxidases and protein engineering, which we've seen for aryl alcohol oxidase and HMFO. But often there's an overlapping substrate scope due to the limity of this homology-based genome mining strategy. But hopefully advances also in protein structure modeling through this advance. The structure of enzymes can be well predicted even in the absence of high sequence similarity to known proteins, which can then facilitate the discovery of novel alcohol oxidation enzymes. Yes, with that, I'm at the end, and thank you for your kind attention.